So hi, I'm Simon Guillot, and I'm second PhD candidate in Dr. Luc Dupuis' lab, and we're going to leave sales for this talk, and we're going to focus on a very different kind of impairment in ALS, and more specifically the orexin-dependent one. So first and foremost, I'd like to introduce one of the major brain regions that we have in the brain that's called the hypothalamus, because it's known to regulate metabolism, sleep, reproduction, circadian rhythm, and much more. And more specifically, here we're interested in the lateral part of the hypothalamus, the LH, where we have dozens of neuronal population, but most importantly, we have the MCH and orexin neurons that are mainly known to regulate sleep. And it's known since recent days that in sporadic cases of ALS, familial cases, and also pre-symptomatic gene carrier of ALS, their total volume of hypothalamus is decreased and significantly decreased compared to healthy control. And moreover, we have TDP-43 aggregates in the lateral part of the hypothalamus, so exactly where the MCH and orexin neurons are. And so in the lab, we wanted to understand whether this TDP-43 aggregate and this atrophy in the hypothalamus was having any impact on this MCH and orexin population. And so what we did is to first assess the MCH expression in two mouse models. So the first one is SOD-G86R, and the second one, the first delta NLS, and both have been assessed at pre-symptomatic stage of the disease. And so we have a clear-cut decrease of the MCH expression in the two mouse model of ALS. But for the orexin expression, the level remained completely unchanged for some reason. So what we know from here is that we have an atrophy of the hypothalamus where we lose the MCH neurons, but the orexin remains unchanged for some real reason, and that the MCH expression induces sleep and the orexin induces wake. So we wanted to decipher the physiological consequence behind this hypothalamic wiring in ALS. So the first experiment we performed was to implant a novel EEG implant in the SOD and the first mice to then record a 24-hour recording continuously coupled with indirect calorimetry, and then assessed all this data that we just recorded. So the first thing we had to look at, because as I mentioned, MCH and orexin neurons are mainly known to regulate sleep, is actually sleep. So the first thing we have is an hypnogram, which will tell you in which sleep stages you are. So either the wake or different sleep stages. And what we can just find by looking at these two hypnograms is that the SOD at pre-symptomatic stage has like straight lines for hours, which is not normal, because those animals are nocturnal animals. So here in this period of time, they're supposed to be sleepy, and here awake. And when we quantify those, is a clear increase of wake compared to the wild types, but also a clear decrease of the REM, the rapid eye movement phase, so the deep sleep, and the REM, the non-rapid eye movement phase, the light sleep, and we have a clear-cut decrease in those two sleep phase at pre-symptomatic stage of the disease. And so we replicated the same experiment in the other mouse model, the first model. So at 3 months of age, I'm not going to spend too much time on here, there is nothing to show, but at 10 months of age, we have the same deficits that we observed in the SOD model. Same as the SOD, clear increase of wake, clear decrease of the REM and the NREM as well. So we have hypothalamic atrophies occurring decades before the symptom onset that leads to sleep deficits occurring also at pre-symptomatic stage in our two ALS mouse model, where we have an increased wake episode and a decreased REM and NREM episode. So as I mentioned, we are losing the MCH neurons, so there's no point of targeting those, they're not there anymore. But we still have the orexin neurons, and we know that they are responsible for wake episodes. So can we target those specific neurons in the LH to reduce the sleep deficit that we observed in our two ALS mouse model? So a very easy way and efficient way to do such is to use a pharmaceutical tool. So one we found is a drug that's already available in the market. It's called Orexin. It's a sedative and hypnotic molecule. It's been approved and is currently in use by the FDA in the US since 2014, still pending since 2016 in the EU. It's a double orexin antagonist, and it's been tested multiple times. It's currently in use in psychiatry and endocrinology to improve sleep, where it shows significant decrease of wake and increase in sleep. However, one of the major drawbacks of that drug is its maximum efficacy of 2 or 4 hours after the injection. So with this drug available, we decided to repeat the same experiments as before, but given the CRBS to do 2 mouse model of ALS. And I'm only showing the SOD just because of time limit, but the same thing happened in the first mouse model. When given the drug, we decreased significantly the wake back to the baseline of the wild types. But not only the wake, but also the sleep, where we increase the REM and the NREM in the mouse model back to the wild types level again. So because I mentioned MCH and Orexin neurons, mainly involved in sleep regulation, we have an effect. We have sleep deficit, but the Orexin can rescue this defect. But they are also known to have a very major role in metabolic regulation. And because I mentioned we had an indirect calorimetry, we had a look at this data, and I'm only going to show you 2 major parameters. First, the body temperature. Here in the back curve, this is the wild types. So when you're sleeping, your body temperature goes down. But again, with the SOD in purple, the temperature does not go down at all. It remains higher than the wild types during the sleep period. But when given a Orexin, we're completely able to rescue the body temperature that was higher before. And another one that's really important as well is respiratory quotient. It's a ratio that will tell you which nutrients your body is using to function. So when you're sleeping, you're not eating, so your body is using fat. Here, the SOD mouse model is using carbohydrates instead of fat. And when given a Orexin, we force the mice to use fat instead of carbohydrates. So we rescue the metabolic deficit given the Orexin as well as the sleep. So in conclusion, we now know that for a fact that there are hypothalamic atrophies occurring in patients before the symptoms onset, and that there is an hypothalamic rewiring arising during the pre-symptomatic state of the disease, where we lose the MCH neurons in both patient and mouse model of ALS, whereas the Orexin neurons are completely preserved in the disease, and that the sleep and metabolic defects that we observe are Orexin-dependent in our 2 mouse model of ALS, but that we can rescue the sleep deficit using the repurposed drug called the Orexin. So as a final thank you, I would like to thank our sponsors, without whom the study would not have been possible, and my 2 mentors and supervisors, Dr. Dupuy and Dr. Barbera, and you for your attention.
Thank you for a nice presentation.
Thank you for your presentation. I have 2 questions to link to the clinical practice. So my first question is in patients. The data you presented in patients about hypothalamic atrophy and TDP-43 pathology, is it end-stage disease? Usually it's autopsy results. And the second question is about sleep disorganization. In patients, it's known to be linked to diaphragmatic dysfunction and nocturnal hypoventilation. So do you have, I know it's difficult in mice, but yeah. Do you have data? Because we know that they develop diaphragmatic dysfunction, we just do not monitor it in practice. So do you have data to link your sleep disorders and diaphragmatic dysfunction and nocturnal hypoventilation?
So for the first one, it was done also on pre-symptomatic gene carrier of ALS. I'm sorry, what was the first one? I forgot.
Do you have data on hypothalamic atrophy at pre-symptomatic stage in human?
Yeah, that was on the slide where we had the sporadic cases, familial, and also the pre-symptomatic gene carrier of ALS. So those were pre-symptomatic cases, and those also depicted an hypothalamic atrophy.
So it's MRI data, I guess?
Yes, yes, those were MRI data, yeah.
Okay.
And for the second one, so all the sleep defects appeared at pre-symptomatic stage, and we currently have collaboration with Holmes Hospital, where they also have pre-symptomatic gene carriers of ALS, and we are still waiting for the data on that.
Okay, and for sleep? Sorry.
Well, for sleep, they seem to have sleep deficits, but after the symptoms onset. Before the symptom onset, it seems likely, but we have to analyze the data, the EEG as well, because it's still ongoing for the humans data.
Okay, thank you.
Thank you.
Thank you. I have one question. If you use the in a chronic way your drug, I don't remember, Orexin.
Yeah.
Oh, okay. Did you observe modification for the weight of your animals?
We only did one chronic injection by gavage. We are planning to do, no, sorry, it wasn't acute, but we are planning to do a chronic assessment on that, given the dose only during the sleep period of time, it's the same as a human would take the drug, to see whether it has an impact on the lifespan of the mice, any impact on metabolism as well. So it's planned, but we didn't have time to first do that.
Thank you.